Hello, my name is Barbara Lowe Fisher. I'm the mother of a son injured by a vaccine in 1980, and this is a commentary brought to you by the nonprofit National Vaccine Information Center. On March 22, 2010, FDA officials adhering to the precautionary principle told American doctors to suspend use of Rotarix vaccine until the agency finds out why DNA from a pig virus was found in that live rotavirus vaccine. The FDA says there's no evidence at this time that the vaccine manufactured by GlaxoSmithKline and given to babies at 2, 4, and 6 months of age to prevent diarrhea poses any safety risk. The discovery that viral DNA is contaminated Rotarix vaccine was made by a team of scientists at an independent research lab in California, where they used new technology to detect fragments of viral genetic material in vaccines. More testing confirmed that many copies of DNA from the pig virus were present in all doses of Rotarix vaccine released since it was licensed in 2008, because the original seed stock used to make the vaccine was contaminated too. This surprising discovery was made after the independent lab used new technology to evaluate the purity of 8 live virus vaccines for polio, rubella, measles, yellow fever, chickenpox, rotavirus, and MMR. In addition to pig viral DNA in Rotarix vaccine, DNA fragments from a bird virus was found in measles vaccine, and DNA fragments of a virus similar to monkey retrovirus was found in Rotatec vaccine. After the team double-checked their findings, they notified GlaxoSmithKline on February 9, who notified the FDA on March 15th, and the FDA suspended use of Rotarix vaccine on March 22nd. The FDA says it does not know how DNA from a pig virus got into Rotarix vaccine or whether it means that intact virus is in the vaccine. They're doing more studies to find out. So how is Rotarix vaccine made? Rotarix is a genetically engineered vaccine that GlaxoSmithKline created by isolating a human rotavirus strain infecting a child in Cincinnati and then using African green monkey kidney cells to produce the viral seed stocks from which all Rotarix vaccine has been made. Because cow serum was used to prepare the seed stock, the vaccine had to be tested to make sure it was not contaminated with mad cow disease, a form of transmissible spongiform encephalopathy, which is a brain-wasting disease. An enzyme from the pancreatic juice of a pig was also used to make the original viral seed stock, and there's some speculation that may be how the pig virus got in there. There's another rotavirus vaccine used in the U.S. called Rotatec. It's a genetically engineered vaccine containing 5 human-cow reassortment strains of rotavirus that were created at the Children's Hospital of Pennsylvania, where strains of rotavirus that give cows diarrhea were combined with strains of rotavirus that cause diarrhea in humans. The reassortment viruses were transported to Merck, where master seeds were produced using African green monkey kidney cell cultures. Fetal cow serum and an enzyme from the pancreatic juice of a pig was also used to make the seed stock. There are small amounts of cow serum and monkey viral DNA that remain in Rotatec vaccine. A February 2010 FDA document listing non-binding recommendations for vaccine makers says vaccines should be free of adventitious agents to meet the FDA requirements for purity, and that residual DNA might be a risk for causing cancer or being infectious if it remains in vaccines. Contamination of vaccines with animal viruses is not new. In the 20th century, polio vaccines given to tens of millions of people worldwide were contaminated with simian virus 40, which was the 40th monkey virus identified in polio vaccines that used monkey kidney tissue cells for production. SV40 was found to cause cancer in animals and is associated with human brain, bone, and lung cancers, although the government denies that those cancers are caused by SV40. There also has been controversy about the link between experimental polio vaccines tested in Africa in the 1950s and 1960s that were contaminated with a monkey virus called simian immunodeficiency virus, or SIV. Soon after the polio vaccine trials in Africa, the human immunodeficiency virus, or HIV, that causes AIDS emerged. Lots of questions remain about the failure of technology to screen for monkey viruses. Vaccine manufacturers have long used cell material that comes from the bodies of mammals, including vaccines in use and experimental vaccines that use cells from humans, monkeys, cows, pigs, dogs, and rodents, as well as birds and insects to make vaccines. There's always the risk of contamination with viruses and other microbes, or DNA from those microbes, that can escape detection. There has even been discussion among vaccine manufacturers and the FDA in the last decade about using cancer cells to make vaccines, but the risk of contamination with cancer cell DNA is a big risk. In searching for ways to make seasonal influenza vaccines in a faster, easier, and less expensive way than relying on chicken eggs for production, drug companies have experimented with dog kidney and human fetal retinal cells. But these cell lines have been documented to cause tumors in animals, especially the dog kidney cells. At a November 19, 2009 FDA Advisory Committee meeting, a vaccine manufacturer asked for permission to use caterpillar cells to make pandemic influenza shots. But insect cells can be contaminated with insect viruses that are hard to detect. The FDA committee on that day voted no. There are lots of questions about how the manufacturer of Rotarix vaccine, as well as the FDA, totally missed identifying pig virus DNA contaminating the original seed stock and all doses of Rotarix vaccine given to more than 1 million American children in the past few years. Is there state-of-the-art screening technology that is being used by private laboratories, but not the drug companies and the FDA? Why did the independent team of scientists who found the contamination notify the vaccine manufacturer first, rather than also immediately reporting directly to the FDA? And what about the significance of finding bird viral DNA in measles vaccine and monkey viral DNA in Rotarix vaccine? The contamination of Rotarix vaccine is only the latest in a long history of vaccine contamination issues that require a re-examination of the way vaccines are made and tested. It's a wake-up call for industry and government. The big question people are asking is, why do drug companies making vaccines continue to use cells from animals, birds, and insects that can be contaminated with viruses and other adventitious agents that are hard to detect? The FDA was right to suspend use of Rotarix vaccine until they know more. Hopefully, this serious vaccine production and testing issue will be addressed immediately by vaccine manufacturers. If not, the next pandemic or serious health problem may be one that comes out of a vaccine lab. For live links to references for this information in this commentary, go to www.nvic.org. Thank you.